Hello students, welcome back. Today we are going to start another topic. Okay, before going through the topic, I would like to discuss that in the previous lecture we were doing chapter 2 biological classification. In this chapter we were discussing about the five kingdom which was given by R.H. Whittaker. Okay, so in this kingdom there are five different kingdom, that is kingdom Monera, Protista, Fungi, Plantae, Animalia, and out of them we have done three kingdoms. What are they? Kingdom Monera, Protista and Fungi. Now the remaining two kingdoms, kingdom Plantae and Animalia, we will discuss in detail in the next chapter, because in your NCERT it is given in the different chapters. Chapter 3rd of kingdom Plantae and the another one chapter 4 for the kingdom Animalia. So now to complete this chapter, we will start with some remaining topics which we will discuss here, that are viruses, viroids, prions, lichens. Why we are discussing these topics in a separate one? Why? What is that reason? Because R.H. Whittaker was not able to classify these organisms. That's why we are studying in a separate one. Okay, so let's start the topic then. First of all, we will start with the viruses. What are the viruses? We all know something in a very short form what viruses they are, neither living nor non-living. Okay, so these are some statements which we know here. So let's start about the study of viruses. When we are saying that scientifically we are studying about any particular organism, so here that are viruses. So that study it is known as virology. Virology is viral relate to virus and logy relates to study here. Okay, the next point it is not truly living. You must have heard about that viruses are the connecting link between living as well as non-living one. Why we are saying this? Because we are not considering viruses as a living as well as non-living, because there are some characteristics in the viruses which are showing these characters. Okay, so that's why it is written here, these are not true living. When they will be living, when it will enter inside any host body, then that time it will become living, otherwise they are non-living in nature. Another point which is shown here, non-cellular. As it is non-living, okay, it may be living or non-living, but when they are non-living, so we know that there will be no particular cellular structure is there. So we are saying it is non-cellular structure, or they are not having any plasma membrane, they are not having any cell organelles. Okay, it is made up of only what? Nucleic acid as well as protein. Okay, in the next one you will come to know that the structure of viruses, or the how viruses are made up of. So here I am writing here nucleoproteins. Okay, what is the meaning of nucleo? Relates to nucleic acid, which is a genetic material, and another one is a protein. Fine. Then another point which I have written here, inert crystalline structure outside the living cell. See, as we are considering these viruses are non-living outside any host body, when they will get any host body, then it will get replicate, otherwise they are present as a inert particles. Okay, just a non-living one. So we have written inert crystalline structure just outside the living cell. Okay, and another point which I have written here, once they infect the cell, they take over the machinery of the host to replicate themselves, killing the host. And we know that as it will enter inside any living cell, what it will do, it will infect that host, and it will form any disease there. Okay, so we can say that what it is happening, there will be some machinery of the host will act upon it, which will replicate these viruses, and these viruses are able to affect any host, or it will create some disease in that particular organism wherever it is going inside the body. See here, these replication can take place inside the plant body. Okay, it may be animal, it can be human. Okay, so viruses can attack anywhere in plant, animal or human. Then another point is intracellular obligate parasite. So same thing, as we are saying, it is infecting any living cell. So what they are doing, it is entering inside any cell and it will become the obligate. It means true parasite. Okay, it is acting upon any, what are the parasite, which takes nutrition and they will affect or they will create some disease into the host body. So what it is happening, it will act as a obligate parasites. As outside the living body, what it is showing, it is showing any non-living nature, or they don't require any oxygen, they don't require any metabolism. So whatever it is happening inside the cell body only, when it will enter inside the host cell. Okay, another important point about the viruses are viruses outside the host. As I have said, the viruses which are present outside any host body, they are non-living in nature. They are in form of some particle. So what we are saying them, virion. Okay, this term is very important. What are the virions? We cannot say these are the viruses. These are the particles which are present outside the host body. Okay, when it will enter inside any living organism, it will become the viruses. Fine. So these two terms are there when you will say viruses and when you will say virion. These two difference between these two terms. Fine. Now here in this topic of viruses, we will study more about the history of viruses and who were the scientists that were related to the discovery of viruses. Fine. So the first name in your NCERT which is given, Pasteur, Louis Pasteur. Okay, so what he did, he gave the term virus, the virus term given by whom? By the Pasteur, but he was not known about the structure of virus. Okay, so what he has done, that on the basis of their nature, that it is affecting some organisms, so he said this virus term means venom or poisonous fluid which is affecting the host body. Okay, so what you will say, Pasteur coined the term virus, which means the venom or poisonous fluid which is able to infect the host cell. Okay, the another scientist which came here, D.J. Ivanovsky, in 1892. What he did, here I am showing some diagram. I have written, recognize certain microbes caused mosaic disease of tobacco. Okay, whenever question will be there, the first virus have been identified where? In the tobacco mosaic virus, TMV. Okay, so the virus was discovered by Ivanovsky in which plant? In the tobacco plant. Okay, and we know that tobacco is a cash crop. So it was grown more and more that time. So this scientist, what he did, for example, he saw the leaves of the tobacco and there will be some yellow patches, and he said some mosaic we can say mosaic microbes are present, but he was not known that it is a virus or any other microbe. So what he did, he crushed the leaves. Okay, he crushed the leaves and take the extraction of the diseased leaves. Okay, so after taking the extraction, what he did, he passed this extraction through the bacterial proof filter. And what are the bacterial proof filter? These filters are those in which bacteria get filtered. Okay, bacteria is not able to pass on through this bacteria proof filter. But when he passed this extraction through this bacteria, I am writing here bacteria proof filter, what happened? Bacteria proof filter, what happened? These microbes which was there, he extracted from these leaves, was passed on through this filter. So he concluded that whatever the microbes which are present in these leaves, which is affecting these leaves, these microbes are very smaller than the bacteria. Okay, bacteria is small in size, but these microbes are still or even very small than these bacteria. This bacteria. So same thing he has written. The recognized certain microbes. He have recognized certain microbes which is present in these leaves, and they are causing some disease in tobacco. Still, he was not able to give more about the viruses. Okay, so if I will ask what Ivanesti did, he extracted the this microbes from the disease plant and passed through the bacteria proof filter, and afterwards what happened that the these microbes get filtered from this bacteria pass filter, and he concluded that finally these are very smaller in size than the bacteria also. Okay, now the third point about the discovery that M.W. Bijurnik in 1898. Now what he did, he took again some fluid from the diseased plant. Okay, if you will see the structure, there will be some patches in the diseased plant of tobacco. So what he did, he took some fluid, and this fluid he passed to the healthy plants, and finally he concluded that if we will take the fluid from the diseased plant and will give to the healthy plant will also get infected from the same disease. Okay, and this fluid name, it is Contagium vivium fluidum. This point is very important. Okay, so in examination it can be like this, that this word given by whom? So it is given by the Bijurnik. This fluid is an infectious fluid. It is having the ability to infect even the healthy plants also. Okay. Now the fourth scientist, again which is very important, Stanley in 1935. What he did, he extracted, or what we can say, he said that viruses could be crystallized, and we know that the non-living material can be crystallized, and this crystallized form is made up of, or we can say it consists of proteins. Okay, so the work of Stanley is what? He demonstrated the crystallization of viruses, and the viruses, these crystallization are made up of protein. Fine. Now we have done the discovery or these important scientists. Now we will discuss about some structure of viruses. We know that viruses having some structure, so how it will look like, as we have done that the first virus which was discovered in the tobacco mosaic virus. So how it will look like. Okay, and how there are different examples, as we know that what are the examples of the disease caused by viruses, like mumps, measles, polio, AIDS. So these are the examples which are causing disease by the viruses. Fine. So let's start with another structure of viruses here. We will start the structure of viruses. Okay, so here as we have discussed that the composition of virus, as it is a non-cellular. Okay, so here it is the structure of virus is made up of nucleic acid as well as the protein code. So two things we will discuss here: genetic material, and the genetic material made up of nucleic acid, and this nucleic acid can be either DNA or RNA. So the genetic constitution of the viruses are DNA or RNA. But you must know that it cannot happen that both will happen or both will present at the same time. Either it will be DNA or it may be RNA. Okay. Now the viruses that infect the plants and animals, as we have said, the virus having the ability to infect the plants as well as animals. So the genetic constitution of the viruses which will infect the plants, that is ssRNA, and this ssRNA is what? Single stranded RNA. And the viruses which are infecting the animals, we are talking about the general. Okay, some exceptions are always there, but the mostly the viruses genetic constitution which will infect the animals, it may be either ssRNA or DNA. It means that it can made up of RNA as well as DNA, or single stranded or as well as double stranded. Okay. Now the another thing is protein code. Okay, so what is protein coat? Protein coat is the that covering which will present outside the genetic material, that is known as capsid. So capsid is what? It is a protective covering which will cover the genetic material of the viruses. And these capsid is made up of what? These capsids are made up of some small subunits, or we can say capsid is a whole, and these are the some small subunit which will form whole this capsid form. So if you will see here the structure which I have drawn here, so this whole structure is capsid and the subunits are known as capsomeres. Capsomeres are what? These are the subunits of the protein coat, and totally it will form the capsid here. Okay, so the thing which I have written here, which arranged in helical or polyhedral geometric forms. So if you will see the protein coat, you can see that these can be present in different forms. It can be present in the helical form, okay, round in structure, or it may be in polyhedral, as you can see in the bacteriophage. So here I have drawn viruses, that is TMV, tobacco mosaic virus, and here I have drawn bacteriophage. Okay. So in TMV you can see that there is a helical structure, and in this bacteriophage you can see some polyhedral form. Fine. So now we will discuss about some more example of the viruses here. If you will see that in this diagram I have shown the general structure. Okay, so what is the meaning of general structure? In which I have shown the outer covering, that is what? It is a nucleocapsid. Here I have shown the nucleocapsid which is made up of protein, and inside this there is a genetic material, and this genetic material can be DNA or it can be RNA. Fine. And outer towards there are some more structure, that is the inner covering and that one is the outer covering. And after the outer covering you can see that these spikes are present, and these envelope protein will act as what? It will act as a structure which will attack on the host cell. Okay, or we can say it will help the virus to attack on the host cell, and this will decide the structure of the viruses also. Fine. So here I have mentioned some examples which are causing diseases in case of animals, or we can say in humans. Okay, HIV, you all know about the HIV virus which is causing AIDS, hepatitis B, influenza, rabies, rotavirus, Ebola virus. Fine. Now another one which I have shown here, that is TMV. TMV is what? As I have said that it is the first virus which was discovered by the Ansky. So it is a tobacco mosaic virus. If you will see the structure of this virus, how it look like, helical structure, and as I have said that this virus will affect the plant, and the virus which is affecting the plant, it is having the genetic material, that is single stranded RNA. So this structure is what? It is a single stranded RNA, and in between you can see that there is a central lumen, and outside that you can see some capsomeres are present, and as I have said, these are what? These are the protein coat. Okay, and total it will form the capsid. Fine. In case of TMV, there are how many capsomeres? For example, it is 1, 2, 3, 4. So if you will count the whole capsomeres in TMV, these are 2130. Okay, so 2130 capsomeres are present in the tobacco mosaic virus. In your NCRT, these two diagrams are given, which is very important from the examination point of view. They can give you the diagram to label it and to explain about the diagram. So here I have drawn the TMV virus, in which we have shown different parts. Fine. Another one, that is bacteriophage. See, virus can affect the plant, it can affect the animals, as well as it can affect the bacteria also, and the viruses which can infect the bacteria, those are known as bacteriophage, and we are saying bacteria eater. Okay, so here I have drawn the structure of bacteriophage. It consists of different parts. The parts are head, which is at the upper part, then collar, then neck, and these are the tail. Okay, and these are the tail fibers. So if first of all, if you will see the structure of bacteriophage, head is there. Head, the outer part, it is made up of capsid. Capsid is what? It is a protein. Okay, and inside this there is a genetic material which is what? It will be the double stranded DNA. You have to memorize that bacteriophage having what type of genetic material? Double stranded DNA. While in case of TMV, what will be the genetic material? It will be ssRNA. Fine. Now another thing, that is collar and then neck, and below to this there will be some tail. Okay, in this tail, in between there is a core, and core is covered or protected by some sheath, and combinedly what we are saying, it will form the tail of the bacteriophage. Now there is a tail fiber, and what is the role of these tail fiber? These tail fiber helps to anchorage. It will help in a anchorage of this bacteriophage into the bacteria. When this will attack in a bacteria, what it will do? It will remove all the structure outside, and only the genetic material will get inside, and it will get replicate. So these tail fibers are useful to attack the bacteria. Okay, now when it will infect the bacteria, what it will do? It will send its genetic material to the bacteria, and there will be replication of the genetic material, and it will form different bacteriophage from the bacteria. Okay, so here we have done all about the viruses. What are the examples of viruses which are causing the animal as well as in the human beings. Along with this, some viruses are there which are causing diseases in case of what? In case of plants. As we have done, one of the diseases, TMV, tobacco mosaic virus, and another disease, it can be in case of banana, bunchy top of banana, in which what happens, the above leaves will form like a bunch, and it will stunt the growth of the banana. And in case of this mosaic, you can see the symptoms. There will be some curling in the leaves, there will be some yellow patches in the leaves. So these are the indication that the plants are affecting with the viral diseases. Okay, so here for today we have done viruses. In the next one we will do viroids, prions and lichens. These are smaller topics which we will cover it in the next lecture. For today, that's all. Thank you.